Hello, welcome to Biology, the scientific study of life. More about protein and nucleic acids. Nucleic acids store and transmit hereditary information. If the primary structure of a polypeptide determines the conformation of a protein, what determines primary structure? The amino acid sequence of a polypeptide is programmed by a unit of inheritance known as a gene. Genes consist of DNA, which is a polymer belonging to the class compounds known as nucleic acids. The roles of nucleic acids. There are two types of nucleic acids, deoxyribonucleic acid, DNA, and ribonucleic acid, RNA. Notice that DNA, which mean the deoxyribonucleic acid, RNA ribonucleic acid. Okay, there are two, both DNA and RNA. These are the nucleoids that enable living organisms to reproduce their complex component from one generation to the next. Unique among molecules, DNA provides directions for its own replication. Molecules, DNA also directs RNA, which means ribonucleic acid, synthesis, and through RNA controls protein synthesis. Notice this diagram. Also, DNA is the genetic material that organisms inherit from their parents. Each chromosome contains one long DNA molecule, usually consisting of from several hundred to more than 1,000 genes. When a cell reproduces itself by dividing, its DNA molecules are copied and passed along from one generation of cells to the next. Encoded in the structure of DNA is the information that programs all the cell's activities. The DNA, however, is not directly involved in running the operations of the cell any more than computer software by itself can print a bank statement or read the barcode on a box of cereal, just as a printer is needed to print out a statement and a scanner is needed to read a barcode. Proteins are required to implement genetic programs, the molecular hardware of the cell, the tools for most biological function consisting of proteins. For example, the oxygen carrier in the blood is the protein hemoglobin. Not the DNA that specifies its structure. How does RNA, the other type of nucleic acid, fit into the flow of genetic information from DNA to proteins? Each gene along the length of a DNA molecule directs the synthesis of a type of RNA called messenger RNA, mRNA, as it's written or as is shown here. mRNA, which means messenger RNA, ribonucleic acid. The mRNA molecule then interacts with the cell's protein synthesizing machinery to direct the production of a polypeptide. We can summarize the flow of genetic information as like this: DNA, RNA, protein. Notice that DNA, RNA, protein. As it's seen here, the actual size of a protein synthesis are cellular structures called ribosomes. In a eukaryotic cell, ribosomes are located. Notice that. Ribosome. Notice. Ribosomes are located in the cytoplasm, but DNA resides in the nucleus. Messenger RNA conveys the genetic instructions for building proteins from the nucleus to the cytoplasm. Prokaryotic cells lack nuclei, but they still use RNA to send a message from the DNA to the ribosomes and other equipment of the cell that translate the coded information into amino acid sequences. The structure of nucleic acids. Nucleic acids are macromolecules that exist as a polymers called polynucleotides. As indicated by the name, each polynucleotide consists of monomers called nucleotides. A nucleotide itself composed of three parts: a nitrogenous, as it's seen here, a nitrogenous base, a pentose, a 5-carbon sugar, notice that, a pentose, and a phosphate group, notice a phosphate group. The proton, the portion of this unit without the phosphate group is called nucleoside. Notice that, nucleoside without a phosphate group, called a nucleoside. Nucleotide monomers. To build a nucleotide, let's first consider the two components of the nucleoside, the nitrogenous base and the sugar. There are two families of nitrogenous base, pyrimidines and purines. A pyrimidine has a 6-membered ring of carbon and nitrogen atoms. The nitrogen atoms tend to take up H positive from solution, which explains the term nitrogenous base. The members of the pyrimidine family are cytosine, which C, and thiomin, T, and uracil U. Purines are larger with a 6-membrane ring fused to 5-membrane ring. The purines are adenine A and guanine G. The specific pyrimidine and purine differ in the function groups attached to the ring. Adenine, guanine, and cytosine are found in both types of nucleic acid. Thiomin is found only in DNA and uracil only in RNA. The pentose connected to the nitrogenous base is ribose in the nucleotide of RNA and deoxyribose in the DNA. Notice that the only difference between these two sugars is that deoxyribose lacks an oxygen atom in the second carbon in the ring, hence its name, because the atoms in both the nitrogenous base and the sugar are numbered. The sugar atoms have a prime, notice, after the number to distinguish them. Those as second carbon in the sugar ring, which mean prime 2 carbon, and the carbon that sticks up from the ring is called the 5 carbon. So far we have built a nucleoside. To complete the construction of a nucleotide, we attach a phosphate group to the 5 carbon of the sugar, and the molecule is now a nucleoside for a monophosphate, better known as a nucleotide. Polymers. We can see how these nucleotides are linked together to build a polynucleotide. Agist nucleotides are joined by covalent bonds called a phosphodiester. Linkage between the OH group on the 3 carbon of one nucleotide and the phosphate 1, the 5 carbon of the next. This bonding results in a backbone with a repeating pattern of sugar phosphate units. The two free ends of the polymers are distinctly different from each other. One end has a phosphate attached to 5 carbon, and the other end has a hydroxyl group and 3 carbon. We refer to these as 5 end and 3 end respectively. We can say that DNA strand has a built-in directionally along its sugar phosphate backbone from 5 to 3, somewhat like a one-way street. All along this sugar phosphate backbone are appendages consisting of the nitrogenous base. The sequence of base along DNA or mRNA polymer is unique for each gene because genes are hundreds to thousands of nucleotides long. The number of possible base sequences is effectively limitless. A gene's meaning to the cell is encoded in specific sequences of 4 DNA bases. For example, when the sequence AGGT means one thing, whereas the sequence CGCTTAACC has a different meaning. Real genes, of course, are much longer. The linear order of base in a gene specifies the amino acid sequence, the primary structure of a protein, which in turn specifies the protein's three-dimensional conformation and function in the cell. The DNA double helix. The RNA molecules of cell consist of a single polypeptide chain like a one shown here. In contrast, cellular DNA molecules have two polynucleotides that spiral around an imaginary axis, forming a double helix as shown here. James Weston and Francis Crick, working at Cambridge University, first proposed the double helix as the three-dimensional structure of DNA in 1953. The two sugar phosphate backbones run in opposite 5 and 3 directions from each other, an arrangement referred to as interparallel, somewhat like a divided highway. The sugar phosphate backbones are on the outside of the helix, and the nitrogenous base are buried in the interior of the helix. The two polynucleotides, or strands as they are called, are held together by hydrogen bonds between the paired bases and by van der Waals interactions between the stacked bases. Most DNA molecules are very long, with thousands or even millions of base pairs connecting the two chains. One long DNA double helix includes many genes, each one particular segment of the molecule. Only certain bases in the double helix are compatible with each other. Adenine A always pairs with thymine T, and guanine G always pairs with cytosine C. If we were to read the sequence of bases along one strand as we traveled the long length of the double helix, we would know the sequence of bases along the other strands if a stretch of one strand has the base sequence like this one, for instance. AGTCC3, then the base pairing rules tell us that the same stretch of the other strands must have the sequence 3TSSAGGS. The two strands of the double helix are complementary, each the predictable counterpart of the other. It is this feature of DNA that makes possible the process in preparation for cell division. Each of the two strands of DNA molecule serves as the template to order nucleotides into a new complementary strands. The result is two identical copies of the original double-stranded DNA molecule, which are then distributed to the two daughter cells. Thus, the structure of the DNA accounts for its function in transmitting genetic information whenever a cell reproduces. Thanks for listening.